back. Can you listen to me? Yes? Shh. Yes. It's working. Welcome back, guys. It's a pleasure to introduce Professor Theresa Ong. Theresa is a biologist by training with her bachelor degree from Williams College in the US. And she earned her master's and PhD degrees in ecology and evolutionary biology from University of Michigan, also in the US. After that, she was a postdoc researcher at Princeton University in the US, and she's currently an assistant professor in environmental studies at Dartmouth College, also in the US. In Theresa's lab, they are interested in the complexity of coupled human-natural systems, including agriculture and cities. Especially, their interest in the ecology of food systems and the interactions between people, the environment, and communities of organisms in agriculture. Oh my God. For this, in her lab, they combine mathematical models with lab and field experiments to understand how food systems respond to social environmental stressors, including climate change and economic recessions. But Theresa is not only interested and restricted to agroecology, she also makes much more than that, and I'm very, very glad that she's going to present today about landscape patterns that emerge from small scale species interactions. So please help me to welcome Theresa.
Thank you so much. Thank you. Hi, everyone. Thank you, Flavia, for the invitation. Can you all hear me okay?
Yes.
All right. So I'm very excited to talk to you about our recent work on coral halo patterns. And first off, I just want to thank my collaborators, Peter Vasconcelos, Luo Junyang, Chunyang Xu, and Lisa McManus, a couple of whom I know will be joining you in a couple of days. So if you have any questions about coral halos and you're excited about this topic, you'll have plenty of people to talk about it with shortly. So let's just dive in, and let's start by just addressing what are coral halos. Okay, so this picture here is Heron Island. It's part of the Great Barrier Reef in Australia. And you can see the coral heads here are actually the grayish color. You can see them sort of dispersed throughout the lagoon. The bluish color is that beautiful tropical water, which indicates that you can see down into the substrate, the sand. And then there's this greenish color that's spread throughout the space, which is actually microalgal mats. But what's particularly striking is that in these satellite imagery, we can observe very interesting patterns, spatial patterns that surround the coral heads. So specifically, if I zoom into part of the reef here, you can see that nearby the coral heads, there are these seemingly patches of empty space, which we call grazing halos. And these grazing halos have been observed actually since the 1960s. And basically what they are an absence of algae. And they seem to be varying in shape, but they also seem to have a fixed width as you move away from the coral head. In the case of Heron Island, the algae here is a microalgal mat. But in other reefs, the vegetation could be seagrass, it could be seaweeds. But what we basically see is some grazing that's happening as you move away from the coral heads. So what's really interesting is that these halo patterns have been observed at a global scale. So you see them in satellite imagery worldwide. And Elizabeth Madden has been leading a recent effort in tracking these coral halos, and she's found them in all parts of the globe. And you can see this picture here from one of her studies showing how ubiquitous these patterns of coral halos seem to be. And though they seem to be of relatively fixed width, they do vary from site to site. And the obvious question is why? Why do we see these patterns? The major hypothesis involves the behavior of the herbivores in the system. So coral reefs are these intricate ecosystems which create spatial refuges for prey that are able to hide from predators within the reefs themselves. And Madden's research group has really demonstrated eloquently that these the grazing pressure of a reef declines the further you go out from the head of the coral. So in this study here by Olivier et al. In 2018, you can see in graph C that we have on the x-axis distance from the patch to the reef in meters. And on the y-axis, that is the algal canopy, so the height of the algae. And you can see that it's pretty low at a certain distance from the reef, and it tends to increase over time. And the leading hypothesis is that it's predators, which live in the open waters and will go to reefs to feed on herbivores. They tend to cause the herbivores to stay close to the coral heads, which means that then there's like a limit in which the herbivores are foraging away from the corals, and it creates this interesting ring pattern that we're seeing. So this hypothesis fits right into the idea in ecology called the landscape of fear. And the landscape of fear is this other hypothesis, this idea that prey can actually alter their behavior in the presence of predators. And this can actually have potential consequences to population dynamics and the spatial patterns of prey that are not due directly to predation, but are actually these non-consumptive behavioral effects that are induced by fear, right? So for example, in the temperate region where we are at Dartmouth, moose may feed less and have higher mortality, lower reproductive capacity, simply because they are afraid of predation, not necessarily directly through the predation events, which do happen as well. But even just the fact that you're afraid of a predator means that you're going to alter your behavior, forage less, and have a consequence for your population. So in the context of coral halos, this application of the landscape of fear hypothesis is particularly interesting because if halos themselves are related to predation, then it is possible that we could use the presence of these coral halos, which are observable from space in these satellite images, as an indicator of reef health, right? So we know that coral reefs are under attack from a variety of different pressures, including overfishing. And when we overfish reefs, we tend to lose first the top predators in the ecosystem. So I don't know if I can ask a question out to the audience, but I guess my question to all of you is, what would you expect to be the case if a reef had an overfished population? What would happen to the halos? Yeah. Well, I can't hear the response, but I assume that you're saying
maybe they would go away.
That the halos would go away. Well, that's interesting, right? Because you can imagine that the halos are being produced by the herbivores. So if the herbivores have no predators, then what would you expect? The herbivores might start consuming more, right? And if they consume more, does that mean there's less halo or more halo?
I guess halos would expand, yes.
Yeah, halos would be expanding, right? That's the idea. But it turns out to be a little bit more complicated than that. We're seeing that these halos don't have a simple explanation, because it is true that Elizabeth Madden has shown some studies that in these protected reefs, we find more halos than we do in areas that are unprotected, meaning that they're protected from fishing these predators. But it's not necessarily completely consistent with this idea that, you know, the halos might grow in size as you have fewer predators. So when I first started, I actually met Elizabeth Madden several years ago now, I think, where we were at a conference talking specifically about the landscape of fear. And when she brought up the system, I found it super fascinating. But I started to think right away about, well, what about the spatial patterns of the corals themselves? What, if anything, does the patterns of the corals have to do with the presence or absence of these rings? So here again is Heron Island, and you can see the distribution of corals throughout the reef is not necessarily random. There are areas that might be random, but there are areas that are more clustered, areas that are more dispersed, and also the pattern of the algae itself is not necessarily random. There's very specific patches that exist in the space. So in this project, we're interested in asking two major questions. The first is, to what extent can the presence of halos be determined through simple geometric rules that relate to the spatial patterning of the corals? And two, are consumer resource interactions necessary to explain the observed halo patterns? So we have a series of methods that we have employed to address these questions, and they basically outline the talk that I'm going to give to you today. So first, we created a geometric model of halos. Second, we created a consumer resource model for halos. Third, we analyzed the actual spatial patterns of halos from the satellite imagery available. And then finally, we started to compare the models in data. So we're still working out bits and pieces of the model, but I will present to you today what we've been working on. And I'm really curious to talk to you more about your thoughts, how we might incorporate those into the model. So curious what you have to think. So I'll start first with the geometric model of halos. And we can start from a pretty basic standpoint and think about a coral as a circle, right? We think about these corals as points in space, and the halos are basically fixed swifts that go out from the coral edges, right? And so geometrically, it's quite simple. It's a series of circles that are overlapping in some cases or not. And that overlap will really depend on where the corals are distributed through the space, right? So we can imagine if I create these landscapes of, say, just L fixed length and width, we see that if we drop our corals in a more dispersed fashion or a random fashion versus a more clustered fashion, we'll get different predictions for how much algae should be existing in the landscape, right? With the more clustered here having more algae. And so very simply speaking, if we think about it from this perspective, the fraction of algae A should be some function of the fraction of coral in the landscape, some function of the halo width H, and lambda, which is the spatial distribution of coral. So step one, how do we define the spatial distribution of coral? Well, we started to use a very simple measure for spatial distributions that was described by Clark and Evans way back in 1954. It's a statistic known as Clark-Evans R. And basically what R is just the actual average nearest neighbor distance, d of n points, divided by the expected average nearest neighbor distance for a Poisson process given row intensity. So we know that in spatial point processes, if you sort of randomly throw points in space, it's not a purely random process. The spatial point process makes it so that things that are closer together tend to be aggregated. And so the null random expectation for a point process is a slightly clustered system. And so if we take the actual measured nearest neighbor distance between points in the landscape divided by the null expectations, then this R value will equal 1 if the distribution that you observe is actually random. It's actually corresponding to that similar Poisson random process. But if that observation of nearest neighbor distances is less than your expectation, then the system is clustered. And if it is greater, bigger distances means that it's more dispersed. So this is a great, very simple statistics that works relatively well in our example here, where we have like a L-squared landscape to look at. And what you can see very simply is that if I take 100 points and I put it in this space here, and I change the average nearest neighbor distance, we move from a hyper-clustered scenario to one that is eventually maximally dispersed. And that also corresponds to increasing R from 0 to 1 to greater than 1 as you move from max clustered to max dispersed. So if we take these theoretical coral heads here, we add some coral halos of a fixed width to them, we can see quite simply that you would expect that in this hyper-clustered scenario, you actually have the maximum amount of algae because of the overlap in halos that occurs when the corals are clustered. And then as you disperse out, you get less and less algae because the halos start to eat up that space as you become more dispersed. So we can take this a step further and see that in this scenario of max packed hexagonal arrangements of the coral heads, you can calculate that the area of that maximally clustered reef should approximately be a circle with radius RA. We're just saying radius RA is here, that radius of all that packing. And then from here, it's pretty simple to see that the fraction of area of algae is just going to be 1 minus the fraction of area of corals, which we estimate here with the area of the circle with radius RA, and also minus then the fraction of area of halos, which is just that RA plus H circle minus the RA circle. And this, of course, simplifies further, but just to give you a full sense of where that estimate comes from. And then in the case where we have the area of, or where the corals themselves are maximally dispersed, we see that that is actually a pretty simple system as well, geometrically speaking, because in that maximally dispersed reef, then the area of algae, the fractional area of algae is simply 1 minus n times each of those individual circles, those fractional area of coral halos minus the fractional area of the halos themselves, right? So we can see it's just like the sum of these smaller circles minus 1. Now, you might expect, of course, then in between the max clustered and dispersed scenarios, the halos overlap. And there are formal equations here that can help us describe the fractional area. But basically, we can very simply in this geometric model sort of predict, given like a specific number of corals and spatial distribution and halo width, what the algae should be. So this is that. Yeah.
Can they make a question during the presentation, or you prefer during after you finish?
Either way, up to you all.
So there is one question coming from Laimara. Go ahead, Laimara.
Hi, it's the graph A. I didn't understand because there are smaller green circles and there's also a green background. The green background is the same color of the algae color in the B graph. So what is the,
Yeah, good question. So I didn't edit this graph. It was for a different sort of reason that we have the different colors, but you can assume that the darker green and the light green are the same in this case. Does that make sense?
But does the light green background is still algae, or the algae can only survive at a certain distance from the coral head?
Yeah, exactly. So that the light green algae and the green algae is actually the same in what we're talking about right now. Yeah, you can ignore the dark green circles. Right now, the dark green circles are only there to show you like the distance between the coral heads. But it's still algae, dark green and light green. Does that make sense?
Yeah, I think you said it's okay.
Yeah, thanks.
Okay, thank you. I can't see. Okay, so that's the scenario here. We have A, which is the maximally dispersed scenario, and B, which is the maximally packed scenario. And we can also simulate random Poisson aggregations of corals to get what you would to calculate how much algae coverage should occur with different halo widths. And basically this is to show you that we can calculate algae, and it should be a function of the number N of the corals, the L, the length of the reef, square reef in this case, RC, which is the radius of the coral itself, and H, the halo width. And in this graph, you can see that we can also sort of fudge this relationship mathematically to make it easier on us by approximating this relationship between halo width and algae with an exponential function, which I haven't graphed here, but it lies in between A and B with certain parameters. Okay, so this is the basis of our geometric model. And then following this, we construct a consumer resource model for halos to include those higher order effects of the herbivores and predators. So to start here, we wanted to think more about the ecology of the system and think more specifically about how algae might grow in a coral reef. And so that, of course, is complicated by what species are there and the different sort of life cycles of different algal species. But in general, there are two modes of which algae can grow. They can grow reproductively in terms of sending out seeds, which will usually land and colonize free space somewhat randomly. And then there's also vegetative growth where an algae can bud from itself and create clonal copies and create an extended network of genetically identical individuals. And so we have at least these two different types of algal growth to account for. And so that is the basis in which we start to think about our consumer resource model. We have an ODE here thinking about the the change in algae fraction, in this case over time. And we say that it should be some function of this vegetative growth and reproductive growth. So starting first with that vegetative growth, we have gamma V here, which is the rate of vegetative growth. And we know that it is times this derivative of algae as a function of H, because we expect geometrically, right, that as the halo increases, algae necessarily decreases, right? And so we also know that the clonal vegetative growth is probably happening at these edges here. And so the bigger halos, they actually have more of that surface area on that edge to grow vegetatively inwards. And then if you have a smaller halo, you actually have less surface area to grow on in the edge here. And then ultimately, when your halos are 0, there's no surface area for the system to grow vegetatively. But in addition to this vegetative growth, and then this vegetative growth here is also multiplied by the halo, which itself is a function of algae, which we formally described in our geometric model. So that's the vegetative growth. We also have this reproductive growth of the algae, which we expect to occur anywhere there is free space. And we know how much free space is available because it's simply A0, which is all the space in the landscape that's not coral, right? So it's 1 minus the fraction of coral is A0, and then minus the amount of algae A that is currently in the landscape. That gives you, at that time point, how much space there is to colonize reproductively. And so that reproduction happens then within the halos. You can think of them as like little spores or seeds colonizing in that space in the halo. And we also then add here to the population change in, or the fractional area of algae rate is also going to be limited by the herbivores. So the fish, for example, but there are other herbivores. For now, I'm just going to think about fish. And so we just have a simple rate here of grazing for the fish population, which consumes the algae. So this rate of consumption then ties back into the fish population, which grows at some rate R, and then is limited by the mortality of the fish, which we let that mortality, now thinking more specifically about the landscape of fear, we start to think, okay, so that mortality of the fish should have some relationship with the halo, which has a relationship with the algae. So in this case here, we can model M, the mortality of fish, to be some function of that halo. And we're working with a mean field model though, and thinking about these spatial dynamics. But in this case, it makes sense because the halo here kind of represents a bigger halo, means that there is more travel distance from the coral for the fish to do. And so that will incur a variety of things, including more predation events, but also through these non-consumptive effects can have an effect of mortality. And so the idea here is that if you have this bigger halo, there's actually going to be the sigmoid relationship with mortality, in which these bigger halos result in more mortality. You can think there's just like more risk of predation as you're moving out from the coral halo to feed. And then these smaller halos then is the reverse, right? A smaller halo, it's easier for you to like very quickly swim out and consume your algae, which mostly grows at the edges anyway, and come back. And so there's less mortality when you have a smaller halo. So we put this all into the model, and yeah, and I think that that's just an explanation here, the bigger halos being more mortality. Okay, so that is very simply then the basis of our consumer resource model for the halos. And we'll move on to the analysis of the actual spatial patterns that we can see in the satellite imagery of the reefs themselves. So we start here by analyzing the spatial patterns. And it's not necessarily as straightforward as you would expect. You have this reef here, and there's actually a lot of this space that you would not expect coral to be able to grow on at all. And so you have to first think about how to define the study site space. And so there is a really interesting and very useful database available to anyone. It's called the Allen Coral Atlas. And they have a global database which includes polygons of different types of coral habitat. And in our case, in Heron Island, where we see these coral halos tends to be in these shallow lagoons. And a shallow lagoon from the Coral Atlas is defined as a fully to semi-enclosed, sheltered, flat-bottom, sediment-dominated lagoon area. And this area is usually shallower than 5 meters approximately. So this is really nice because it means that we can constrain our analysis to a relatively consistent habitat, which in our case is this shallow lagoon layer. And so they have really nice polygons of the shallow lagoon layers across the globe. And so we took that for Heron Island specifically. And we took this image and overlaid the polygon for shallow lagoon and then extracted that layer. And you can see that that's this right here. It's the only part of Heron Island that is that shallow lagoon part of the landscape. And you can see here we have the corals, we also have the algae, and we have the sand. And this part of the project was led by my graduate student, Crispy. And what she did here was she took the image here of the shallow lagoon, and she classified the benthic layers into the coral, the sand, and the algae. And this is a pretty high resolution image. It's about 1.67 meters squared resolution. So each of the pixels is that area in size. And the Coral Atlas actually defines these different types of benthic types. Interestingly, coral habitat is defined as coral/algae, and that's really interesting. I wonder if anyone in the audience knows why that might be the case.
Anyone? No, I guess. There is one guess here. They are passing the mic. Just a second.
Because of maybe, I don't know, symbiosis between species of corals and algae?
Yeah, exactly. Corals are usually symbiotic with algae, and so it's actually very difficult to determine through just these spectral bands of the satellite imagery. The difference between coral and algae is actually quite hard because they are usually part of each other. So even in the Coral Atlas, that's how they do a rough calculation of all these benthic types, the coral, the sand, and the algae, but it's they say it's coral/algae because they know that there are mistakes there due to the symbiosis. But Crispy, she led the classification of our specific study site. We were able to use some AI techniques a little bit to further classify and train the classification so that we can see the distinction between the coral and the algae and the sand, which are laid out here for you. Okay, so from here, we have this nice data set now of these points, which are 1.67 meters squared in size. And we wanted to start to analyze the spatial patterns of the corals themselves. But the problem here, of course, is that we need to consider the spatial scale in which to do the analysis. And so here I have these plotted in different colors now, but it's all of our points in space. And you can see what we can do is first take this whole landscape and we can throw a grid over the entire Heron Island landscape. And we can decide what the size of our squarish samples should be. But it could have a direct impact on what the spatial clustering of the corals are. And so I've just given you two examples here of different sized tiles that we can create over the landscape, and different examples of what that would look like in terms of the coral patterns. What is nice about doing this is that we can sort of distribute the samples regularly throughout the space and look at the whole sort of available space to us. And when we put down the grid, we can select only the samples that are full square samples. And this helps us in a couple of ways. First, our model is created with a default of a square landscape. And so that's nice to compare to our model results. But also biologically, it helps us to reduce some of these edge effects that you might expect to happen if you're sort of close to the edge of the habitat available to you. That will definitely have an effect to some extent on the distribution of species within that space. This allows us to reduce the edge effects if we take these square samples, and also makes it easier to compare to the model results. So we'll start to move into that final piece here and think, how does the model and the data sort of correspond to one another? And if you remember, I said that we could very simply sort of use a geometric model by approximating it with an exponential function. And that helps us with the data analysis a bit here. So this is just an example of one spatial scale of analysis, the 188 meters squared scale. And you can see the different letters here correspond to where they are on the graph in the left. And it goes from more clustered to more dispersed. You move towards the right, and you can see what that actually means in terms of what those square samples look like in the actual reef. So that's kind of interesting here. You see this effect on the spatial patterns, and we also see a coordinate effect on the fraction of algae, which we can calculate directly from the samples itself. And it does decline. So as you get more dispersed, we see that indeed the fraction of algae does decline. And then as you get more clustered, there's a little bit more variance here to account for, but you do get the highest levels of algae in landscape tend to be where the system is more clustered from perspective of this Clark-Evans R statistic. So this is just one spatial scale. And if you've done spatial analyses before, you know that spatial scale has a really important effect on whether things are clustered or dispersed and patterns of that nature. And so we wanted to make sure that this result was consistent across different spatial scales of analysis. And so we just created, again, these tiles of different sizes and did a similar analysis at multiple scales. And so that's this graph here that I'm showing you, which a lot more dots here. In the legend, the number is 160 by 80 here just represents the amount of squares on the X versus the Y axis. So that higher, the highest number on the legend there is the smallest spatial scale and just increases in size. So with that exponential function, we can actually fit the data to that exponential function and estimate through maximum likelihood fits what the halo width should be for each of these different spatial scale resolutions. And then I have plotted here the means of that fit with the 95% quartiles. And you see they overlap across the spatial scales. So that's nice. It seems like it's consistent across these different spatial scales. The estimates all tend to be about a halo size of 3-ish pixels, which if we account for the actual spatial resolution corresponds to a halo width of approximately 5 meters. So that's very interesting because we can go back into that study that I talked about in the beginning of this talk with Olivier et al. In 2018. And you can see actually that this study was done on Heron Island. So this is where they measured the grazing pressure. And they show here that that grazing pressure starts to decline approximately around 5 meters. So that is really cool. It's like pretty consistent then what we're measuring in these satellite imagery and this exponential approximation of the geometric function, what the halo width is in Heron Island. And that does seem to be consistent across these different spatial scales of analysis, which is exciting to us. And then what's also really cool is that the geometric model does predict this decline in fractionality as you increase in dispersion of the corals. But it doesn't do as great of a job of predicting what's going on in this more clustered space, right? Because what we find definitely here, there's this really concentrated cluster of points where you have low amounts of algae despite having very clustered corals. And so we take this as an indication that the geometric model is insufficient at explaining, especially these values here. And so there might be some consumer resource interactions that are necessary to explain these specific regions where you have high clustering of corals and low levels of algae. Unfortunately, yeah.
There is a question. No, you're not going to make the question. So sorry. There was a question. There is no more question.
Okay, no worries. Unfortunately, we are still sort of working out. Our consumer resource model is being tweaked, and we are still sort of analyzing the results a bit. But it does very much seem like the consumer resource dynamics is capable of alternating between fixed points and limit cycles for the populations of herbivores and algae. And so just really very generally, there is a sense that these population dynamics are likely to occur in particular parameter regions. And that can also mean then that we would expect the halos to shrink and grow periodically. So that's kind of like this interesting idea that we might expect those halos to shrink and grow if consumer resource interactions are at play. And that is interesting because through conversations with Elizabeth Madden, we have heard about this phenomenon of perhaps these halos being able to breathe. This idea that the halos might be sort of like shrinking and growing periodically over time. And you can kind of see this in historical images of reefs through satellite imagery, although the quality of images really depends on location and time period. This is Heron Island. Again, you can see some of that green algae and the corals. And so I'm just showing you just like a time series of different years. And you can see there's definitely fluctuation over time of where those halos are. So that's pretty cool and exciting, this idea that we might be able to link the cycling in the algae halos with the population dynamics of the herbivores as well. Okay, so
Theresa,
some basic, yes, any questions?
there's a question. Are you going to make the question, Laimara? Now there's a question.
Okay.
Could you go back to the slide with the graph with multiple points?
Before?
This one? Or
The one before. Yes, this one. It's because I thought you answered my question, but then I'm not sure. There's clustering in that first part from like 0 to 1 in the Clark-Evans-R. There's like a clustering there in the down part, I don't know. But there's also, I think, much more variation in this first half of the graph than in the second half of the graph, and I wanted to know if you also have explanations for that.
Yeah, that is a really good question too. I sort of glossed over that, but definitely there is this like hump shape of higher points and much more variation. And we haven't quite worked out whether this exponential approximation is going to be what we're finally going to settle on for the geometric model. It gets a little complicated when the halos overlap, but we are still working on a version of the geometric model that will more accurately place the R statistic within our geometric model. And so it could be that when we do that, it will account for this bump. It could be that it won't. In the case that it doesn't, then I think we would have to presume that it can't be the geometric model alone, that it has to do something with those consumer resource interactions, which seem to be most important in this clustered coral scenario, right? And so that's pretty exciting. It gives us at least like this, it narrows the scope in which we might expect to find the importance of the landscape of fear and different things like that. And then in terms of the variation, it really kind of depends on what happens with our consumer resource model. Because here, for example, we can have parameters that will predict unstable cycling versus stable limit cycles, or stable nodes or unstable nodes. And it could just be that in this parameter space of clustered corals that we have much richer dynamical systems properties that account for that higher variation in the data. And I think what's really exciting is this like clustering down here. It kind of either, to me, it could represent an alternative state, for example, like where you expect more extinction of algae versus like a higher level. That is possible, but we need to think about whether that happens. We need to test the model to see if it happens in the model. But from the data itself, it kind of looks like it could be an alternative state there. But it also could just be a sign that you have increasing cycles, right? With these points representing just a snapshot in time where you're really just saying like where you are in the cycle. And so that could be that variation, but consistently low ones means like that's where your bound is. And that makes sense with low values of algae. Does that answer your question?
Says yes.
Okay,
Yes,
great.
thanks.
All right. So exciting ideas here with this drinking and growing of the halos. But in just like a very broad sense, our conclusions from the work that we've done so far, it does seem like the halo width measurements are relatively consistent across the samples that we take at these multiple spatial scales. And it seems to correspond to a halo width of approximately 5 meters, which is also consistent with the studies that Elizabeth Madden's research group has done in Heron Island. And so we feel fairly confident about this halo width at Heron Island being around 5 meters. And it does seem that our geometric rules can accurately predict that there will be less algae when corals are more spatially dispersed. And that's particularly exciting. We can say, like, if you have dispersed corals, you're not going to have algae. And so you can't necessarily conclude anything about herbivore interactions with predators in those cases, because it can be explained just by these geometric rules. But at the same time, the geometric rules alone, they are not sufficient to explain some of the values. They are, however, also pretty good at predicting that there is a possibility of more algae when the spatial, when the corals are spatially clustered, that there should be possibility of more algae. But exactly how much algae, whether that algae is a high or low value, I think here is the role for consumer resource interactions in our model to help explain where the data actually deviates from these geometric expectations. But we're still working on that model, and really curious about all of your thoughts about how we can rethink that, because there's some issues right now with the way we have it formulated. But in addition to all of this, we're very excited also about this idea then of consumer resource interactions resulting population dynamics, which would consequently cause breathing in the halos as well, and that these are sort of consistent with observations in the field of changes in the halo patterns over time. And so a lot of future work possible, I think, out from this kind of study. So with that, I think I'll just stop here and thank you all for listening. Special thanks to Lana and Daniel, who helped us out with original sort of analyses related to this when we were really just starting to think about this project. Flavia for the invitation, all of you for listening. And I'll take any questions.
Oh, thank you so much. I think we have time for a bunch of questions. Go ahead. Can you see them, Theresa? I will try to focus on them.
Yes.
Our camera is very high quality, but they are here, I can tell you.
Theresa, thank you for the very interesting talk. I really enjoyed how you combined the geometrical model with the consumer resource model. And you had some very interesting insights. But you mentioned the landscape of fear, and you modeled the mortality based on the radius of the halos, right? And I was wondering if in the scenario of the landscape of fear, have you tried modeling the grazing rate as a function of the radius of the halo? How does that look like?
Well, so that is one idea. One idea is, instead of having the halo be a function, like having mortality be a function of the halo, that we should make the consumption a function of the halo. And that is definitely a possibility. I think if we did that, we'd have to create sort of a reverse type 3 functional response there, where you would expect the consumption to decline as you increase the halo. The problem that we're struggling with is that we're modeling algae fraction and not necessarily algal individuals. And so it leads to some mathematical complications that have got us confused at the moment.
I see. Thank you. And one further question. Can the measurement of the period of the contraction expansion of the halos help us measure some of the parameters of the model?
Yeah, we hope so in the future, that if once we have like the consumer resource model fully fleshed out, we expect that there will be some interesting insights there, because you could, for example, think about the intervals between cycles being able to be parameterized by data to some extent. And maybe that could tell us more about the health of the populations. But there's still a lot to unpack there in terms of whether, there should be effects of like the community composition on the contraction and expansion of the halos. I think those could be interesting things that could come out from that.
All right, thank you.
There is one more question. While they are passing the mic, there's one question from Zoom. Aline Shimada asks about the coral biology. She would like to know if there is a relationship between the health of coral and coral head and the algae cover or the halos. For example, is it possible to observe smaller halos around bleached corals? Is there any relationship?
Yeah, that's a really good question. We, right now in our geometric model, we just say that corals are static, right? We're just saying, it's weird because I can see myself twice in the image. And there's a lag. But anyway, so that right now we're just presuming that halos are sort of just static elements. I mean, corals are just static elements in the landscape, but you're right that corals are dying, right? And so it's not completely obvious right now to me, like how we would incorporate that, because there's still substrate there for life to some extent, but it's definitely less effective as a food resource, as a cover, as a nesting site. And so we could definitely add that as another layer of complexity. What happens to the corals themselves if they're allowed to degrade over time? And I think that would definitely affect our results.
Great. We have one question from people that are here.
Hello, thanks for the talk. So I was thinking about the corals as if they were a network, and like the corals were nodes from this network, and they have links between them if like one coral is next to another. So I was thinking like if a new node is inserted, is like appears in this network, does he kind of have like a preference to attach to another node that have more links?
Yeah, I mean, that's a great question. When we were thinking about the reef and the herbivores, we were thinking that it's probably likely that the herbivores do swim far enough that they could access all of that space. So it complicates us a little bit when you're thinking about the network, but it's definitely true that there will be like fish that have preferences for particular nodes. And one idea is to go beyond the consumer resource model, which right now just has the herbivores and the algae, to include predators in the model. And in that case, we were thinking we would need to add another element of space. And it could be done through this network approach, but also one idea is to consider doing a spatially explicit model where we're thinking of these halos in a Turing sense, where you have reaction diffusion processes happening with the diffusion of the fish who are going out and consuming the algae and the reaction of the predators coming in. And so I do think that that is like this third level that could be applied to explain maybe something that the consumer resource model might not be able to explain as it is.
Okay, thank you.
Okay, there's one more question here.
Sorry. Okay, I will. It has some relationship to all of the other ones, and I was thinking if about the time scale that you were talking about when you were talking about like this breathing of the halos, because I was thinking like, is this halo dependent? Sorry, I'm trying to think how to formulate the question, but it's relating to time scale. So, for instance, if new corals are allowed to appear, and it has some relation with the last question about like disappearance of nodes near other clustered nodes, but if nodes or, for instance, corals are allowed to appear or disappear in this population, does this time scale is not proportional, but is on the same time scale as this breathing of the halos? And if these halos are there, they persist after corals die, but not if, like, for instance, they're bleached, but the structure is still there, so it's just like the skeleton of the coral, and this halo persists. And if there's like a new node that isn't a coral, just a changing the substrate, so just, for example, I don't know, a rock or something like that, if a halo is allowed to appear around that, or if it's dependent only on like having a coral ecosystem. I don't know if this question is like too much or too broad, but yeah, I was thinking about that.
Yeah, I mean, those are really great questions. And I think that, there's just so many different ways to look at the system. I do think this question of timescales is really important and could be incorporated in our framework right now, because essentially we do kind of have that, because like the corals themselves are static, but we could just give them some kind of level of dynamics, but increase the time scale of which those dynamics occur. Because I think you're right? The rate at which the corals are being affected is going to be quite distinct from the algae interactions, and also perhaps quite distinct from the herbivore life cycle as well as the predators. And so all of these add additional lag effects into the system that could help create that variation that we're seeing in the data for highly clustered scenarios.
Okay, thank you.
Hi, I was thinking about like this process of the halo breathing, and I was thinking if have you checked if if it's possible that instead of consumer resource dynamics, this could be related to abiotic processes and seasonality?
Yeah, for sure, that is a possibility. We do have a term in our consumer resource model, which includes environmental stochasticity, and certainly you can usually force consumer resource interactions into like stability. Often when you add these stochastic environmental variables, but like a seasonal forcing could definitely change the amplitude or frequency of cycles to some extent or another, and it could replace it as well, theoretically, right? We could instead of modeling consumer resource interactions, we could just model seasonality in say some abiotic factor, like you're saying, and it could certainly explain growing and shrinking in algae to explain that. But I do think you would still have these geometric rules applied to them, and perhaps then you could distinguish what is the consumer resource interaction difference between that which would happen maybe as a function of the populations of the fish versus a function of time. You know, maybe you could separate those two. And then I noticed Lisa commented, when Lisa's part of the project, she mentioned that the halos also tend to appear around rocks and concrete blocks. So experimentally, I think a lot of marine ecologists have done these interesting experiments where they place fake reefs and see what happens. But I think it's complicated, right? Because it's also just a sort of substrate that fish tend to accumulate around. And so the difference between an artificial reef and a reef functionally for the fish probably depends, right? So I don't know.
Thanks.
There's one more there. Yeah.
Hi, Theresa. Thank you for the talk. It was very interesting. My question is about the geometric model, where you analyze the relation between the clustering and the fraction of algae. I was thinking, if the size of the clusters on the area of the clusters increase or are high in these grid units, can we have like higher clustering values and low fraction of algae just because of the size of this cluster, or is this accounted on the model somehow? Because maybe this will explain that concentration of low algae fraction on high cluster grids.
You mean like the number of corals being a factor?
Yeah, that's it. The number of corals in the cluster, yes.
Yeah. So that is interesting. It is sort of hard to tease apart with our statistic. I think that we could definitely we do account for it. It's in the model explicitly, the number of corals. And so it's still the case, at least in the data, that is where you see. Well, I guess your point is that the geometric model, we could scale it with the number of corals. The pattern overall is consistent. When you have different values of n, it's still going to have that exponential form. You can reduce the sort of slope of that relationship by affecting the number of coral, but we still expect that to some extent. I think it could be easily accounted for in the data. And that is why we need to work on our exponential equation a bit, I think, because we could easily incorporate that value of the initial fraction of corals to account for that.
Yeah, thank you.
More questions, guys? There's one there.
Hi, Theresa.
Yeah, raise your hand. Just, yeah.
Hello.
She's there.
My name is Melina. So I was thinking about the role of the herbivory in this story, because there is a general assumption in the literature that herbivores consume algae, and because of this, they help corals by controlling the algae populations. But recently, there are many studies showing that the herbivores seems to be much more interested in eating microbial mats than the algae itself. So the herbivore may not be so important as previous talked, at least here in the Southwestern Atlantic. And so I wanted to know if you have any empirical observations or experiments showing that the herbivores are actually playing this role of controlling, like doing this top-down control of the algae. I don't know if it's clear.
Yeah, thank you for that question. I think so. You're talking specifically about herbivores controlling algae on the coral reefs themselves and preventing them from like basically being overgrown with algae and dying out. I think in this case it's slightly different in the sense that the herbivores are consuming algae on the substrate out from the coral heads. And in our system, I haven't done the experiments, but Elizabeth Madden's research group has done those experiments to show that grazing pressure does sort of increase as you go closer and closer to the corals themselves. But I think you're right in saying that there are different types of corals and different types of herbivores, and that could have a different maybe impact on the coral health, which is currently not part of the model at all. But yeah, I would love to do experiments like that, but I'm not a marine ecologist.
Lisa, do you have an idea? Just because I know Lisa and Theresa very well, I'm asking her if she has any comments on that. Lisa is not around, maybe? Yeah.
Oh, no, sorry. Sorry, could you hear me?
Yes.
Hi, everyone. I'll see you in a few days, I guess. Yeah, I guess I'd be curious to know what herbivores the student brought up specifically. But I mean, yeah, so Liz Maiden's work that Theresa was saying, like they literally put chunks of algae, like they put them kind of like on a string, and then so they do observe them directly eating the algae. So I think, but there are so many kinds of herbivores on reefs, right? There's the scrapers, the grazers, the browsers. So perhaps you might be referring to one type that targets microbial mets, but certainly some of the functional groups will go for these like fleshier macroalgae or even seagrasses. So yeah, hopefully that was helpful.
I think, yeah,
Yes,
go ahead,
thank
Melina. Yes. Any last question, guys? No. I think they're a little bit tired of carnival, guys. I'm so sorry for that. We are very happy, by the way, but also sorry for being so a little bit lazy. But I think that's it. I would turn my camera on to say, oh my God, I need to replace pins here. It's so confusing, sorry. Okay, so I'm back. I would like to thank Theresa for being available and accepting my invitation for this talk. It was a very amazing talk, a very amazing seminar. I'm very glad you made it, and I hope you can be here in the next edition. So help me to welcome her, to thank her.
Thank you so much.